Okay, it looks like it's time to get started. Does everybody have audio and video from me? Can I get a thumbs up or indicate in chat if you do, please? Oh, looks like I don't have no. Okay, good, thank you. So a couple things before we get started today. Please remember that quiz number 24 will be online by about noon today. Also remember that next week is the final exam. It's going to be, in fact, let me bring up the syllabus to show you. So the final exam is this coming Wednesday, December 9th at 8 a.m. Okay, please remember that. So we'll be starting an hour earlier than usual Wednesday morning. Okay, so the exam will be from 8 a.m. until 9:50 a.m. Okay, now remember the final exam is going to be 80 multiple choice questions rather than 40. The final exams were 200 points rather than 100. It is a comprehensive final, so we're going to be tested on everything that we covered the whole semester. So as far as study guides go, your best study guide are your old exams. So you should have access to all of your old exams now. Again, let me remind you, this doesn't mean that the questions are going to be the same as you had on your old exams. Okay, some might be the same, some might have the answers switched around. Some of the questions might be changed a little bit, so the answer is different. And some of the questions are going to be brand new questions as well. All right. But so when you're studying your old tests, try to understand what I'm asking. Just don't try to memorize questions. That won't get you as far as trying to understand what I'm asking. And I think that's about it as far as announcements go. So today obviously is our last lecture. So the material that I covered today is going to be the last material that you'll be responsible for on the final exam. So let's get back to the PowerPoint presentation. All right, so when we left off on Wednesday, we were talking about the human heart. So over here on the right-hand side, we have a diagram of the inside of a human heart. So remember, the upper chambers are called atria. So we have the left atrium here, right atrium here. So remember, because we're looking at the anterior view of a human heart, left and right is going to be switched. We're talking about the specimens left and right, and not your left and right. Okay, so the right atrium is actually on the left atrium on the right. We have two lower chambers of ventricles, okay, right ventricle, left ventricle. And separating the two ventricles, we have this area here called the septum. Okay, so when we left off on Wednesday, we were just about to start talking about the events in a heart contraction. So embedded in the upper part of the right atrium, there is the sinoatrial node, or the SA node. Okay, so this is a group of cells that occasionally initiate an electrical impulse. And we can see the electrical impulse indicated as the red dot in this animation. Okay, the impulse from the sinoatrial node travels through the walls of the atria. So this is going to actually, it's shown going through the right atrium, but this electrical impulse is actually going to travel through both of the atria to the atrioventricular node. So the atrioventricular node is another bundle of cells in the lower part of the right atrium here. Okay. Now, as this electrical impulse travels through the atria, that's going to cause them to contract. Okay, so we can see that here. So in this diagram, sinoatrial node is here, atrioventricular node is here. Okay, so again, occasionally the sinoatrial node is going to initiate an electrical impulse that's going to travel through the atria to the atrioventricular node, and this electrical impulse is going to cause the atria to contract. Okay, when the atria contract, they're going to move the blood that they've collected from the atria into the two lower chambers or the ventricles. Okay, and we can see that happening here. Okay, after that electrical impulse has traveled from the sinoatrial node through the atria to the atrioventricular node, there's going to be a pause, a delay of about 0.1 seconds. This just allows the atria to finish pumping the blood into the ventricles. Okay, so again, we're all familiar with the thump, 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 thump rhythm of a beating heart. So after that first thump, there is just the slightest pause, just about a tenth of a second, and that just allows the atria to finish contracting and to finish moving blood into the ventricles. Okay, then from the atrioventricular node, the impulse is carried to the AV bundle, which is located in the septum of the heart. Again, the septum is this wall that separates the left and the right ventricles. The AV bundle is branched and it carries the impulse into the ventricles. Okay, so again, a contraction is initiated by cells in the sinoatrial node. An electrical impulse travels through the atria to the atrioventricular node. And then we have a pause of about 0.1 seconds so that the atria can finish moving blood into the ventricles. Then the electrical impulse is going to continue through the atrioventricular bundle, or the AV bundle, that goes through the septum and then branches into the walls of the ventricles. So this is going to cause the ventricles to contract. So this causes contraction of the left and the right ventricles. And the ventricles then pump blood to all parts of the body. So remember that the ventricles have thicker, more muscular walls than those of the atria. Okay, that's because the atria only have to move blood from the upper chambers to the lower chambers. The ventricles are going to move blood throughout the whole body. Okay, so blood in the left ventricle is going to go through the aorta, through the systemic circuit, throughout the whole body. Blood from the right ventricle is going to go out the pulmonary artery to the lungs so that this blood can get oxygenated, and it, of course, is going to return to the heart through the left atrium. Okay, so some terminology. The systole is the contraction phase of the heart. It lasts about 0.4 seconds. And then we have the diastole. The diastole is the relaxation phase of the heart, and this also lasts about 0.4 seconds. So these terms you probably, even though you might not recognize them, there's something that you might be familiar with. If you've ever gotten your blood pressure taken in the doctor's office, they give you a number for your blood pressure that's a ratio. So there's one number over another number. What that is your systolic pressure over your diastolic pressure. So in other words, your blood pressure during the contraction phase of your heart over the blood pressure for the relaxation phase of the heart. Okay, so again, this is probably something that you are familiar with, even though you don't recognize it. So we have systole, contraction phase of the heart, um, diastole, the relaxation phase of the heart. They both last about 4/10 of a second. Okay, pulse. Pulse is defined as heartbeats per minute. It's about 75 beats per minute in a normal human. There's going to be a lot of variation in this. People who aren't particularly in good shape and don't exercise might have a pulse that's in the 90s. If you're an elite athletes sometimes have pulses that are in the 50s. Average, usually average people, it's about 75 is a little high. I'd say average is about 72 or so. Okay, if you get down into the 60s and 50s, usually we're talking about either an elite athlete or somebody who has a problem with their heart where it beats too slow. If we get above about 100 beats per minute for a resting heart rate, that again, that's going to indicate some kind of a problem. That's too high of a heart rate. So we're probably dealing with somebody who's in poor physical condition if it's above about 100. Okay, but normal, I would say normal pulses in the 70s. I think about 72 is normal. Okay, so I've been using this animation. Actually, this is a really cool animation. It shows what happens during the systole and the diastole, and how the blood is moved from the atria to the ventricles and then through the systemic and pulmonary circuits. It does go kind of fast. If you want to slow it down and have a look at what's happening, you can follow this link. I'll check this out after class. I hope this isn't a dead link. I did add this to the PowerPoint last semester or last year? It was last year. I'll check it out again after class to make sure it's not a dead link. But if you go to the link in the player on Reddit, you can slow down the animation so that you can see a little bit better exactly what's happening during the systole and the diastole. All right, so the last thing we're going to talk about in this chapter is blood. So we also talked about blood in the tissues chapter. So remember that blood is a specialized connective tissue. Like all connective tissues, we have relatively few cells and lots of extracellular matrix. Okay, of course, with the exception of adipose tissue. The types of cells that make a blood tissue, we have erythrocytes and leukocytes. Okay, erythrocytes are red blood cells. Leukocytes are white blood cells. About 99% of the cells in blood are going to be erythrocytes or red blood cells. About 1% are white blood cells. Okay. In humans, the total blood volume is 6 to 8% of the total weight of the body. That's 4 to 5 quarts per 8 to 10 pints. All right, so actually, it's probably a little less than you would expect. 4 to 5 quarts really isn't all that much. One example I like to use is if anybody's seen the movie Reservoir Dogs. There's the scene all throughout the movie. We see Mr. Orange lying there on the docks, in a pool of blood. Okay, the amount of blood that he's lying in is more than what's in a human body. Okay, he would have been dead 10 times over before the end of that movie just from blood loss, if all that blood actually came out of his body. Okay, so the total blood volume, and I see there's a little bit of a typo there. Sorry about that. The total blood volume is only about 4 to 5 quarts or 8 to 10 pints, which isn't an enormous amount of blood. Okay, the components of blood. So just like all connective tissues, we have relatively few cells, lots of extracellular matrix. The extracellular matrix that makes up blood is a liquid called plasma. Okay. Plasma is mostly water, but it also carries nutrients, hormones, which of course are the chemical messengers of the body, ions, dissolved gases. So we have dissolved oxygen, dissolved CO2, and urea. Okay, so again, urea is how we fix our nitrogen waste in the blood. And of course, this urea is going to be filtered out in the kidneys. That we talked all about that in chapter 40. A protein that's abundant in the plasma is a protein called serum albumin. Okay, this functions as an osmotic counterbalance, keeps the blood plasma in osmotic equilibrium with the body's cells. So obviously in our blood cells, particularly in our red blood cells, they're just packed full of protein, right? There's this protein called hemoglobin that binds oxygen, carries oxygen from the lungs to the tissues of your body so that you can do aerobic respiration. Okay. Now, if we didn't have serum albumin to counterbalance all the proteins inside of the red blood cells, that would mean the red blood cells would be in a hypotonic environment. A hypotonic environment is bad because water is going to want to go down its concentration gradient. Okay, that means into the red blood cells. So the red blood cells would swell up and could possibly burst. So we need a counterbalance. We need to put enough protein in the serum to make it isotonic to the inside of the blood cells. Okay, so that's what serum albumin does. It provides an isotonic environment for the blood cells so that they don't swell up and possibly burst. Okay, so like I said, blood is composed of two types of cells. We have red blood cells or erythrocytes. There are about 5 million red blood cells per milliliter of blood. They have a very unusual shape. I'm sure during the tissues lab, everybody had a chance to look at red blood cells under the microscope. So they have this very unusual shape called a biconcave disc. So they're like a flattened disc with little dimples in either side. Okay, very small in size as well. So if we look at an average animal cell, we're talking about 20 micrometers in diameter or so. 20 to 30 micrometers in diameter is about average for an animal cell. Red blood cells, however, are about 6 to 8 micrometers in diameter. So small as far as animal cells go. They contain hemoglobin, which binds O2. So again, they're absolutely packed full of this protein called hemoglobin, which binds oxygen in the lungs and then carries it to the tissues of the body so that we can do aerobic respiration. Mature red blood cells have no nucleus. Okay, now this is true for humans, okay? Not necessarily true for even all mammals, okay? There are some other species that have nucleated red blood cells. However, in humans, mature red blood cells do not have a nucleus. And again, I'm sure you were able to observe this under the microscope during the tissues lab. They only last for about 120 days. After 120 days, red blood cells that are worn out are recycled in the spleen, or they could be consumed by cells of the immune system, like macrophages. New red blood cells are produced in the bone marrow. So in the bone marrow, I'm gonna try to write with my my cheap Chinese mouse pen. It doesn't always work out real well. But in the bone marrow, particularly in the red bone marrow, which we only really find in the sternum and the hip bones. Only a couple bones have the kind of bone marrow that make blood cells. We have these types of cells called, I'm never going to get through this, pluripotent. You know what? I'm going to give up on this. And I'm just going to go ahead and use the whiteboard. Okay, so we have these types of blood cells called pluripotent hematopoietic stem cells. Okay, so this works a little better than trying to write with my my cheap Chinese mouse pen. So inside the bone marrow, we have these types of cells called pluripotent hematopoietic stem cells. Pluripotent hematopoietic stem cells make all types of blood cells. Okay, so they make red blood cells, they make all of the different types of white blood cells, they make platelets, and they make they make megakaryocytes, which are the cells that make platelets as well. Okay, so we have pluripotent hematopoietic stem cells. They reside in the bone marrow, and they're responsible for making all types of blood cells. All right, so here's a diagram of a red blood cell. So again, they have this very unusual shape called a biconcave disc shape, small in size. So I said they're about 6 to 8 micrometers in diameter, so rather small. And they have this unusual biconcave disc shape. So if we look at it on the side view, we can see where the biconcave disc comes from. We have these little dimples on either side. Okay, this is actually pretty important because remember in the last Wednesday's lecture, I talked about capillaries. So you know how capillaries are the smallest diameter blood vessels, and they can get so small that only one red blood cell can squeeze through at a time. So this biconcave disc shape makes them flexible enough that they can kind of bend and kind of wiggle through these very small blood vessels. Also, there are carbohydrates on the surface of the red blood cells. Carbohydrates are negatively charged. They're very hydrophilic, so they're kind of slippery. Okay, the carbohydrates on the surface kind of lubricate red blood cells, and that also is going to help them to get through very small capillaries where the diameter is so small that only one blood vessel can pass through at a given time. So again, carbohydrates or polysaccharides are on the surface of the red blood cell. Again, this helps to lubricate the red blood cells so they can get through the very small capillaries. But this is also what's responsible for ABO blood typing. All right, so rather than trying to draw on the PowerPoint, let me go back and use my whiteboard again. Sorry. Okay, so there are two types of polysaccharides that are on the surface of a red blood cell. We have one type of polysaccharide that's called the A antigen, and another that's called the B antigen. Okay, so we have A and B antigens. Let's consider 4 red blood cells here. So let's say we have 4 red blood cells from 4 different individuals. Let's say that the red blood cell over here on your far left contains the A antigen. I'm just going to write that like the letter A. Okay, so individual number 1 has the A antigen. So I just wrote that as the letter A on the surface. Individual 2 has the B antigen. Okay, so I just wrote that like the letter B on the cell. So A antigen, B antigen. Let's say individual 3 has both the A and the B antigen. Okay. And then let's say this person over here, individual number 4, has neither the A or the B antigen. Okay, so this is where we get the ABO blood typing from. So if you only have the A antigen on the surface of your red blood cells, you have blood type A. If you only have the B antigen on the surface of your red blood cells, you have blood type AB. I'm sorry, you have blood type B. Okay. If you have both the A and the B antigens, you have type AB. And if your blood cells lack the A and the B antigens, that means that you have blood type O. Okay. So again, these A and B antigens are polysaccharides that are found on the surface of the red blood cells. If you have the A antigen, you have blood type A. If you have the B antigen, you have blood type B. If your blood cells have both the A and the B antigens, you have blood type AB. And if your blood cells lack both the A and the B antigens, you have blood type O. Okay. Okay, so here's a little bit of a better picture of red blood cells. So we can see that biconcave disc shape pretty clearly. Again, relatively small cells, generally about 6 to 8 micrometers in diameter. Okay, so tiny, and having this unusual biconcave disc shape. All right, then we have the white blood cells, or leukocytes. So again, most of the blood cells that we have are going to be red blood cells or erythrocytes, about 99%. About 1% are going to be white blood cells. There are several different types of white blood cells, and they function in your immune system. Of course, your immune system is going to attack any foreign invaders to try to keep any pathogens out of your blood. Just like the red blood cells, these are produced by stem cells in the bone marrow. These are the pluripotent hematopoietic stem cells that I was talking about earlier. Okay, they make all types of blood cells are made in the bone marrow by these special types of stem cells. Okay, so you might be wondering if these are white blood cells, why are they purple here? That's because they're stained so that we can see them. Cells don't show up really well under the microscope unless we stain them to make them more visible. So we can see a couple different types of red blood cells here, or white blood cells rather. You don't have to be able to identify the different types, but just as an example, here and here, Uh, these are a type of white blood cell known as a neutrophil. Okay, neutrophils are phagocytes, so these can actually engulf organisms by phagocytosis and digest them to get rid of them. So you might remember that macrophages are also phagocytes. You might remember that animation I showed you of a macrophage consuming a mold spore. This one right here, this is a lymphocyte. Okay, there are two types of lymphocytes. There's T cells and B cells. Okay, it's impossible to tell whether this is a T cell or a B cell just by looking at it. You'd have to do further experiments to figure it out. This is a type of white blood cell over here called an eosinophil. Okay, eosinophils, we usually see these in immune responses to parasites. This is a basophil, and this is a monocyte. Okay, so again, you guys don't need to do this, but I'm just trying to illustrate for you, some of the different types of white blood cells that we see. So this is actually a very unusual picture in that we have 5 different types of white blood cells all together in one picture. It's really unusual to see that. In case you're wondering, this little thing here, that's a platelet. Okay, platelets aren't actually white blood cells. They're actually fragments of cells that are involved in blood clotting. Okay, but really nice picture of some different types of white blood cells. Platelets. So I just mentioned platelets. Pardon me, platelets. So like I just said, platelets actually aren't cells. They're fragments of cells. They're derived from large cells in the bone marrow called megakaryocytes. Okay. Now, megakaryocytes, just like all of the other types of blood cells, are derived from the stem cells, the pluripotent hematopoietic stem cells in the bone marrow. However, unlike the other types of blood cells, megakaryocytes don't leave the bone marrow. They stay in the bone marrow, and their job is to make platelets. Okay, platelets are small cell fragments that bud off of the surface of the megakaryocytes. Okay, and the function of the platelets are involved in blood clotting. So blood clotting actually is something very important. If our blood didn't clot, we would completely bleed out before a wound would heal. So it's important for blood to clot so that we can prevent too much blood loss. Okay, so here's another picture. So what we're looking at here, this is a scanning electron micrograph, or an SEM. We can tell that by the nice 3D effect that we get. This one's been colorized so that we can better visualize the different types of blood cells. So we can see some erythrocytes or red blood cells here, okay, with the biconcave disc shape. Here are some white blood cells. Okay, without these white blood cells being stained, we really can't tell what type of white blood cell they are. And then these little fragments down here. Okay, again, these aren't cells. These are fragments of cells called platelets. Okay, and they're involved in blood clotting. All right, so looking at the different types of blood cells. So of course we have red blood cells. These are the most abundant, so about 99% of all of the blood cells in your blood are red blood cells. Again, so there's just a couple types of bones that have the red marrow that can make blood cells. We see that in the sternum, also in the hip bones, in the ilium, and in the femur as well. They contain these pluripotent hematopoietic stem cells. Okay, these are the progenitors of all types of blood cells. So pluripotent hematopoietic stem cells make red blood cells. they make macrophages, they make lymphocytes, and they make megakaryocytes. Okay, megakaryocytes are going to stay in the bone marrow, and they make platelets, okay, which are the fragments of cells involved in blood clotting. Okay, different types of white blood cells. So we have white blood cells are referred to as leukocytes. So we have eosinophils, neutrophils, basophils. Of the types of white blood cells, neutrophils by far are the most abundant. Okay, again, neutrophils are a type of white blood cell called phagocytes. These can actually, engulf and digest pathogens that invade the body. We have macrophages. So in that diagram, we saw immature macrophages are called monocytes. So we saw a monocyte in that picture that I showed you a few slides ago. Mature macrophages, however, we don't see these in circulating blood. These reside in your tissues, okay, and they're constantly patrolling your tissues, looking for anything that's out of the ordinary, anything that's not that shouldn't be in your body. Okay, and again, the macrophages are also phagocytes. So when they encounter something, they will engulf it and then they will digest it. Okay. Then we have the lymphocytes. So again, we saw a lymphocyte in that picture that I showed you a couple slides ago. They look very similar. It's actually impossible to tell a B lymphocyte from a T lymphocyte just by looking under the microscope. We have to do experiments to figure out exactly what proteins are present on the surface in order to classify it as a B lymphocyte or a T lymphocyte. Okay. These are involved in antibody responses. So these make antibodies. Antibodies are proteins of the immune system that are very specifically sticky. So they will stick specifically to determinants on the surface of pathogens called antigens. So on the surface, let's just say this is a bacterial cell down here. Okay. This bacterial cell has proteins on its surface, and these antibodies can recognize and bind to the proteins on the surface of the bacteria. Okay, and there's several ways that your immune system can use these antibodies to get rid of the bacteria. Okay, so what I'm drawing here, an antibody actually looks like kind of like the letter Y, and the sticky part of the antibody is kind of like at the ends of the arm of the Y. Okay, so you can remember this, if you remember like doing the YMCA dance, where your hands are, if you're making a Y, where your hands are, these are the sticky parts of the antibody. They actually stick to pathogens or foreign invaders in the body. Okay, so B lymphocytes and T lymphocytes, these are involved in antibody responses. T lymphocytes are also responsible for cytotoxic responses. So sometimes if your cells are infected with a virus, or sometimes there are some bacteria that invade cells, when things are inside of cells, they're hiding from our immune system. So we need to actually kill the cell that's hosting the virus or the pathogen. Okay, T lymphocytes do that. They come by and they give that cell a signal to undergo programmed cell death. So you might remember apoptosis or programmed cell death from the development chapter. So it's basically, it just gives it a signal to tell it to undergo programmed cell death, and that cell will just kind of fold up and die in a very orderly manner. All right. So that brings us, I believe, yeah, that's the last slide of this chapter. We've only got a couple of minutes left in the semester, actually. So I don't think we should start the immunology chapter. It's unfortunate that we didn't get to it, but as you guys know, it's been a crazy semester. Um, and we just have to accept the fact that we're not going to get to everything that we would like to get to. So the circulation chapter is the last one that you'll be responsible for on the final exam coming up on Wednesday, 8 a.m. Remember, 8 a.m. rather than 9 a.m. when we usually start. So unless there's any questions, let me just remind you one last time, quiz number 24 will be online about noon. Remember, if you're trying to calculate your grade, remember that I haven't dropped your 4 lowest quiz grades yet. It's just much easier to do that for me once all the quizzes have been taken, and I can import that into an Excel file, and then program that Excel file to automatically identify the lowest quiz. It's much easier to do it that way, especially when you're dealing with like 80 students. About how long did diastole last? It's about 0.4 seconds, 0.4 of a second. Okay. Okay, so that's quiz number 24. Also remember, next week, Wednesday, 8 a.m. Is the final exam for this course. Okay, and it's worth 200 points. 80 questions worth 2.5 points a piece, plus a 10-point bonus question. And other than that, if there's any questions, thanks for a good semester. I mean, you guys, I know it's been tough on everybody. It's certainly been a very hard semester for me. I know it's not been any easier for you guys, but thanks for bearing with me. You got through it. If there's no questions, I will end the meeting here, and hopefully I'll see you guys around campus next semester. So enjoy your winter break. Good luck during finals week, and I will see you next semester.